Hello, welcome and thank you for joining us for this webinar looking at the current status of biofungicide use in protected edible cropping and their integration into IPM programs. The speakers will discuss the drivers for using biofungicide products, the challenges that growers face integrating them into the crop protection programs, and potential solutions. I'm Natalie Key, Knowledge Exchange Manager for AHDB Horticulture, and I'll be hosting you today. So before we kick off, I just wanted to run through some housekeeping elements and then order of play for the webinar. So for information, throughout the webinar, the audience is muted, but you can ask questions anonymously by submitting them through the questions box, which should be on the panel on the right-hand side of your screen. You'll have an opportunity to ask questions after each session and at the end of the webinar, and I'll read these out. It would be helpful if you could indicate who the question is for, just for ease. And if there are specific concerns or questions you have as a grower relating to biofungicide use that could be fed into how we develop future research and trials, please also submit those and I will feed them forward. Any questions we don't get to will be saved and answered after the webinar where possible. And if there are follow-up questions following the webinar, please email them to me on natalie.key@ahdb.org.uk. Basis and Enroso are also available for this webinar. So if you did not submit your details when you registered to claim these, you can send your details via the questions box or to Aaron or myself. Right, sorry, I've just lost my way. So the webinar will be recorded and will be made available on the website after the event. So for the order of play, we'll start off with Ant from Fargrow, who will provide an overview of where the protected edibles industry is with the use of biofungicides and highlight potential new advances and opportunities to increase efficacy and adoption. So I will pass over to Ant. Over to you, Ant. Thank you.
Great. Let me just share my presentation. Okay, hopefully everyone's got that. Great. Perfect. That's the first hurdle out of the way. Yeah, so my name's Ant Surrage. I'm a technical development manager at Fargrow, and today I'm just going to be giving an overview of the state of play for biopesticides, specifically biofungicides in protected edible cropping. So I want to look at four main key areas. Firstly, the drivers for change. So what are the forces that are either pushing growers away from conventional chemistry or pulling them towards more biopesticide usage? The barriers to adoption that we're hearing back from growers in terms of the issues with efficacy or technical issues around application of product. How we can look at overcoming those barriers through creating robust holistic IPM programs or through the use of new application technology or smart technologies, and then touch on the potential pipelines of products that could be coming through to the UK. So if we start off on the drivers for change, the way I see it, there's two different sets of drivers. The push factors. So I think these factors are relatively familiar to growers. Stuff like loss of synthetic chemistry. So we're seeing a lot of the active ingredients when they came up for review under 1107/2009 not being authorized again. And unfortunately, the approval of biopesticides, although good at the time, wasn't necessarily providing like-for-like replacements for that synthetic chemistry, which ultimately meant that there were gaps being formed in growers' toolboxes. Alongside that, issues with resistance, so overuse of certain active ingredients leading to the selection of resistant populations of diseases, which of course is further exacerbated by loss of synthetic chemistry. As you start reducing down the active ingredient pool, of course, there's going to be greater selection pressure on the ones that remain. And finally, legislation and societal pressure. So legislation, of course, fits in with that loss of synthetic chemistry, but there's further legislative pressures such as the Sustainable Use Directive and also the new National Action Plan that are trying to push growers away from the use of conventional chemistry. And of course, societal pressures. We live in an information age where information is freely available to the consumer Whether or not that information is particularly balanced, many would argue not so, but it certainly creates a demand from the consumer base, which is ultimately fed through to people like the supermarkets around reduction in usage of pesticides. But there are still some pull factors pulling growers towards the use of biological controls and biopesticides and biofungicides. Firstly, the increased investment and development of alternative technologies. So a lot of smaller spin-off companies have proved that the route from conception of a active ingredient as a strain through to commercialization as a product is possible and now defined, which means that investment in these technologies is easier. And on top of that, big ag is starting to take more of an interest in developing biological solutions, which should ultimately mean that UK growers are seeing more and more of these solutions coming through to market. On top of that, there's also an increased investment in technologies to improve the biopesticides themselves' efficacy. So a great example of this, of course, is the AMBER project and the work being done by the researchers alongside Silso to improve the application of these products, as well as the use of smart technologies to improve them, which I'll touch on slightly later. In terms of the barriers, I see there being three key barriers at the moment to the broad-scale adoption of these products. Firstly, efficacy. So of course, biopesticides tend to have lower intrinsic efficacy than conventional chemistry, where you might have had a conventional chemical that has a control rate of 85, 90%. A lot of biopesticides would be around that 60 to 70%. And then tied very closely to that is the predictability of that efficacy. So it might work really well one time, but then the next time you use it, less well. And I say they're so intrinsically tied together because if you can consistently and predictably obtain that 60%, 70% efficacy, it forms a base from which you can start building your IPM program. So I know Rob Jacobson mentions it very often that you have that first line of defense and then that second line of defense. And going forward, we'll have a third and fourth to give the overall program the same level of efficacy, if not a better level of efficacy than the conventional chemistry did have. But all of that leads to the third issue, which is ultimately complexity. So the understandings of these systems is very complex, it's very knowledge intensive, which ultimately means in the peak of the season when growers are particularly busy, it's very difficult to go through all these different individual decisions, look at the complex interactions between different products and disease development and try and make the right decisions. So it's about how we can reduce that down. So in terms of how we can overcome those barriers, of course, one of the most important and fundamental points is training and understanding. So sessions like these or sessions led by the AHDB or by us on knowledge exchange, working with growers to exchange knowledge to make sure they have the best understanding possible. But probably as important and some of the best groups I've been part of in the last couple of years have been grower interaction groups where we have growers who are using these products, coming together, talking to each other, sharing their experiences to move forward. Application, which I will only touch on quickly because I think there'll be more from Dave later on this. But the need for greater understanding of application of how to get these individual products to work best, especially when they perhaps need to be applied to areas of the plant that are more difficult to reach with conventional methods. Formulation, so the potential to formulate biopesticides in new and innovative ways to perhaps give them more UV protection or protection from the environment in general, to give them a bit more longevity on the leaf, or the potential with bioinsecticides especially to have some form of attractant in there that's going to help the insect come towards it and feed on it. A whole systems understanding is going to be important moving forward. So understanding how all these different products interact. So if you're using organic fertilizers, for example, or biostimulants, what sort of effect does that have on the biopesticide you're using? Is trichoderma using that to multiply in the soil, and is that improving its efficacy? Things like that are areas that are really going to be of interest in the future. And the use of smart technology. So I'll touch on smart technology in a bit more detail because it's one of my key interests. So at the moment, we're seeing growers use smart technologies to make better data-driven decisions. So a lot of this data they'll already be capturing through their climate control systems or perhaps remote monitoring systems. And they can use this data in two key ways as I see it. Firstly, to ensure that the biopesticide or biofungicide is always being applied at the right time, so ensuring that the environmental conditions are not going to be a limiting factor. And you can set up dashboards that will present the data back to the grower in an easy to view format, giving instant insight. And then the second point would be to track and predict disease progression. So again, you can present that data back when you know the thresholds for certain disease developments to tell you you're starting to get more at risk. And why is this important in my view is because if we are applying the product in the right conditions at the right time in the disease progression, then we start to answer that question around predictability, because we're always applying it at the right time, which should give us good efficacy. And it also starts to take away that question of complexity. We're taking the complexity out of the hands of the people on the ground and putting it in the cloud. So it's processed there and just presented to the growers. So in the peak of the season, you can very quickly action insights. But I'm also going forward, there'll be lots of new technologies that are helping growers make these decisions. So I just thought I'd touch on two quickly for pre-symptomatic disease detection. One that I think people will be more familiar with, which is hyperspectral imagery. So using hyperspectral cameras to get reflectance readings from leaves that then give a certain signature that are telling the algorithm that there's something wrong with the plant, which ultimately means the grower can get out there and treat pre-symptomatically, so before they can see the disease. And that will mean that we'll get better usage out of some of the biopesticides that are more early curative than preventative, because they don't last particularly long in the environment. It will mean we can be really precise with the timing of those applications. And then the technology on the right-hand side in these pictures, which growers might be slightly less familiar with, which are sap flow sensors and stem diameter measurers. And they're measuring the differences diurnally in the diameter of the stem, as well as the sap flow through the plant. And this is working in a similar sort of way as electrophysiology would, in that when you have a disease come in, you see a marked difference in that sap flow and stem diameter because the plant's under considerable stress. And that can be an early indicator of there being something wrong. Still in its early stages in terms of disease prediction, but an excellent product in terms of setting a firm base for your IPM program and ensuring you have a robust plant. Ultimately, it's important that these decision-making systems are easily automated in terms of the spraying. So these systems are giving actionable insight back to the growers. So it's great to say, yeah, you've got a problem, but the solution from that problem needs to be actionable in the sense that growers have to have those products on site or a way of applying it. A particular issue is when we work in outdoor crops around whether you have a spray window, but in terms of protected cropping, if you don't have the product there on site, it's all well and good saying you should use it, but it's not there, and by the time you get it, the conditions might not be appropriate. So there's a lot of work to think about how we can ensure that when we have these suggestions, we have the products ready to use. And then ultimately, all folding into robotics, which I'll touch on in the next slide in slightly more detail. So there are still some clear potholes in the pathways to having this successful. Firstly, better understanding of various pests and diseases. So there is quite a lot of lab-based studies and some field studies on disease progression. However, when you get into real-world commercial cropping, there's issues that come up that aren't really thought about in experimental procedures. So very simple example, that sensor on the right-hand side, an infrared arm. In experiments, there'd probably be someone daily out there checking it, still pointing at the leaf. In the real world, it gets knocked about, might end up pointing at that piece of wood or at the sky, and then you start getting readings that are affecting the prediction. So there's ways of how can we make sure that that's not going to happen. Technical solutions for actioning the insight, as I'd previously mentioned, but very importantly, grower confidence and understanding. So there is the potential that we could start having these issues with a digital divide, where we have lots of new technologies being brought on stream very quickly. And if sensor and technology companies alongside suppliers aren't working shoulder to shoulder with growers to make sure there's understanding and to make sure that the products are being developed in a way that's actually useful for them, they're never going to see as good a value as they may have if they were developed in tandem. So it's really important from our point of view to make sure we're getting grower feedback at every stage of development with these products to ensure that they're going to be useful in the field. There will be some regulatory restraints around the use of new technologies, and CRD have a whole new set of challenges to focus on with these new technologies, especially with spot treatments and variable rate spraying. And one of the key questions from a grower perspective is always the return on investment. Hyperspectral imagery, new sensor technologies, they tend to be quite expensive, especially in the early stages. However, the easiest way to start getting that quicker return on investment is that these robotic systems end up being multi-purpose. So if you have a system that's out picking, which is going to have a relatively good return on investment because it's cutting down on labour so significantly, but also will have a hyperspectral camera on it to detect fruit at the ripeness stage, that same hyperspectral camera could potentially be used to spot powdery mildew or thrips or downy mildew, and then ultimately perhaps have a mechanism to try and treat those in hot spots, which ultimately means it's multi-purpose and you have a quicker return on investment. So if we move on from technologies to new products that we see coming through or trends over the recent years, if we look at some of the data from the EU Commission on new active ingredients that have been approved since 1996, we see that there's being more and more biopesticides making up the overall number of applications for active substances. And that's reflected as well in the number of different forms of biopesticides that are making up that percentage. So you have more basic substances, more pheromones, more plant extracts, more microorganisms. So what that means is that you have more products overall, but also a greater variety of those products being brought to market. And that active ingredient increase is reflected in the number of products that come through in the UK. So we see that reflecting the active ingredient increase, more and more products coming to the UK. And of course, the first question that will be on a lot of people's minds at the moment is, how is leaving the EU going to affect the products being brought to the UK? And in the short term, we don't see it affecting it much at all, in the sense that a lot of the products that will be approved this year and potentially next year will have been submitted before the 1st of December, and as such will follow the same regulatory time frame and protocols as they would have. Also, the regulatory framework has just been lifted and shifted into UK law, so it's still the same sort of trials and sort of data sets that need to be generated, and the manufacturers we work with still have a very good appetite to bring products to the UK. It's a good market that's established with biopesticides and biofungicides, so they're very keen to continue bringing products over here. There will be some finer points that need to be ironed out as we move forward, but hopefully CRD will have the opportunity to be more pragmatic with some of their approvals, but we'll touch on some of the potential changes slightly later on. So if we look at the pipelines and the potential to expand our horizons, just to touch on a few new technologies that are out there that might be coming to the UK in the coming years. Stuff like bacteriophages are very interesting, so the potential for very specific control of certain bacterial diseases. So if we look at companies like Omnilytics and their product Agriphage, these phages are chemically attracted to certain bacteria. They then attach onto the outside, insert their DNA and take over the cells to reproduce themselves, highly specific to individual bacteria and exist naturally in the environment. So a really unique and sustainable solution for some key bacterial diseases. We think biostimulants will play a particularly important part in bolstering an IPM program. So from the point of view of setting a firmer foundation in terms of a healthier, more actively growing plant, but also the potential for biostimulants to almost act as adjuvants to biopesticides, whether it be as a feeding source through humification enzymes helping to break stuff down in the soil for trichodermas to feed on, or just increasing plants' natural resistance to diseases and helping improve the efficacy of some biofungicides. Plant extracts is a key area as well. So again, if we look at the US with products like Regalia based off of giant knotweed extract, the ability to upregulate certain pathogenosis related pathways within plants to upregulate systemically acquired resistance, so increase the oxygenated species within the plants, increase the production of phytoalexins to help actively fight back against incoming diseases based off of a genetic change in the plant. Specifically with insecticides, the potential for venom-based products is really interesting. So again, in the US, you've got products like Spear T, based specifically off of selected spider venoms that give highly specific controls to certain forms of flies. But I think possibly most importantly, moving forward, is our increased sort of focus on looking at different markets to the ones we've been looking at in the previous years. Obviously, being part of the EU meant that it was very much easier once a product was on, an active ingredient was on Annex 1 or a product was approved elsewhere in the EU, to mutually recognise that product to the UK or use a zonal glasshouse recognition to bring it from the south of Europe into our glasshouses in the UK. But now that we don't necessarily have that advantage, we can potentially look into wider areas. So the US I've mentioned many times here, and the US has something ridiculous like 550 approved biopesticides. And some of that technology is really novel and interesting, and will be really good to try and discuss more with these companies to bring them over to the UK. It's not necessarily that we couldn't do that before, but it's more the sort of shake up in thinking that we've now got around, let's give it a go, let's see what technologies they've got to bring over. And also looking at new total solutions, so finding solutions that improve the overall effectiveness of a program. So if you think about maybe adjuvants and attractives, or attractants and biostimulants, to potentially bolster the overall program efficacy to give you that similar level of efficacy that you might have had with a conventional chemistry program. But there are still some potholes, as there were with the technologies, with these new products coming over. So the best pathway to commercialization is still unclear for R&D companies, although it is becoming clearer as more products make it through the approval process. It's easier to outline to an investment company what the process is going to look like and when they'll eventually start seeing return on investment. But it's still perhaps unclear what companies to work with in certain markets to get a good idea of what the actual market uptake might look like. And there's certainly some questions around the pesticide usage data and how regularly it's updated, and how substantial a market prediction or review companies can do to give an informed decision to investors about what the market size might actually be. But even once you get it through the regulatory processes and to market, the uptake of said products is unclear. I think it will be easier than it has been in the past, with more biopesticides having been uptaken. There's now a better understanding of how stuff like entomopathogenic funguses or just biopesticides in general work. But still, novel technologies such as the phages or resistance-inducing products, the mode of action again is different to that of the biopesticides that we have seen in the past. So there still is a potential that the uptake might not be as ready. So that's an issue in terms of bringing these products to market commercially. So there's currently no distinct regulatory process for biopesticides, which does mean that the biopesticides are being reviewed under a similar framework as the traditional chemistry. There is the potential there for CRD to look at the possibility of perhaps incentivizing the applications and the approvals of biological controls and biofungicides, which would ultimately mean that the UK growers would have a wider portfolio to select from. There is the potential to look at systems similar to that of the US, where they have a government-funded organisation called IR4, who will run some of the trials for the data packages that are needed to get approvals. And that would take some of the cost out of the hands of the sort of smaller spin-off companies that are producing unique strains for unique markets, and again ultimately mean that more products are coming through for UK growers. But at the moment it is a long, expensive and onerous process to bring some of these products through to the UK, which ultimately means a lot of cost, which means that has to be reflected somewhere down the sales process, which is worrying for niche cropping sectors in particular. So if you look at it purely from a commercial point of view, of if you have a particular strain of fungus that's very specific to a certain disease on a specific crop, the market size is quite small, which makes the justification for investment in getting through all these authorization processes quite difficult. And potentially there is the potential there for regulators to be more pragmatic in their decisions and understanding that some of these markets are valuable to the UK, but actually the return that the crop protection company might see on producing some of these unique biopesticides won't be very big, and it might take a considerable number of years for them to see a return on investment. So is there a potential way they can look at working around that? So that gives a general overview of the sort of things I was thinking. I think we're looking at having some questions at the end of the session, or possibly now if there are any that's come through now.
Yeah, there have a couple have come through. Thank you very much, Ant. One question. One issue for biopesticides is cost. Is there any indication that the UK CRD will introduce a lower requirement or lower cost registration regime for these products?
Yeah, so I kind of mentioned it a bit there at the end, and this is a key issue, that the costs for biopesticides at both the grower level and from a regulatory point of view are quite high. There is the potential to make some changes in CRD that would mean that the approach to regulation of low-risk substances and biopesticides could ultimately cost less. I think it's going to be a case of there being need to be industry-wide lobbying from both grower bases and from suppliers and distributors to try and make the case that, especially in some niche sectors in UK horticulture, there is the need for these products, but the commercials just don't work out. So I know CRD are talking about things like this, and it's in discussion at the moment, although they have plenty to deal with at the moment with leaving the EU. So potentially further down the line, there is the possibility for more reform, and we could have systems that are more similar to that of the Environmental Protection Agency in the US and end up having the same sort of numbers of biopesticides being brought to the market. Although there are some downsides of their system, but it's all very much up in the air at the moment.
Yeah, and the next question actually touches on that in a way as well, in terms of the new products described in your pipelines slide. Would that still fall within the remit of CRD?
Yeah, I mean, anything plant protection based is going to fall in the remit of CRD, and I think we'd want all of the products that were, well, we would want all of the products that were claiming to have effects on pests or disease to be registered as plant protection products, because that not only is the regulatory framework at the moment, but also gives you guys as growers confidence that it's gone through all this various efficacy testing and also safety to the end user and environmental safety testing, because that still is absolutely key. So they'd all go through CRD. Yeah.
Okay, I think if there are any further questions for Ant, please do submit them, but we can come back to them at the end. Thank you very much. I'll now pass on to Simon from Vitagress to provide a grower's perspective on trialling biofungicides in his crop protection programme, some of the challenges he's faced, as well as the potential for opportunities for improvement of use of these products. So over to you, Simon. Has that worked?
I hope so. Can you see that? Yes,
we can. Thank you.
Okay. Right, thank you, Nat. Okay, so yes, I'm Simon Budge. I am agronomist at Botrytis Herbs. We're protected herb growers down on the south coast, West Sussex. So just wanted to give you a brief talk about our specific experiences with using biofungicides on this site. My get out clause, as always, is they are very much tailored to how our crop and how we grow that crop and how we apply these products. So other people may have other experiences, but certainly talking to other herb growers, they have had similar results from using biofungicides. I'd love to be able to say they work. I'd love to be able to say how useful they are, but unfortunately, that is not our experience. And I'm going to talk a bit towards the end about perhaps how we can look at ways to improve their use. Ant's already talked about sort of why we want to use these things, and just to reiterate, we want to use less chemical pesticides. We are seeing less and less actives available, particularly in protected herbs, and we're seeing resistance to a number of products developing. And we want to be able to integrate them into our integrated pest management strategy. That's particularly important with biofungicides because, like a lot of growers, we use things like Bavaria bassiana for control of insect pests, and obviously you don't want to be spraying around chemical fungicides when you're trying to use a fungus to control pests. So I'm just going to talk sort of briefly about the products that we've particularly looked at. And really it's these 4 different products here. So you've got the 2 bacterial formulations at the top there, and then 2 fungal formulations. So with the bacterial ones, as far as my understanding goes, both the Serenade and Amlo-X, these products are actually kind of like in 2 halves, in that you've got one part of the product is the polypeptide that's produced by the bacteria during fermentation for the production of the final product. And when you're spraying this formulation onto your plants, that polypeptide is sitting there on the leaf, and any spores from fungi that come along, any mycelium that's growing, they'll then hopefully destroy those spores and that mycelium. You've then got the other half of the product, which is the bacteria itself. So the bacillus, the two different bacillus species there, and they'll again land on the leaf surface, and they'll hopefully actively grow, and they'll have various modes of action, but particularly competition and antagonism against other fungal pathogens that might be there. That's all good in theory, and if you do things like test for presence of these two bugs, you can find them very easily through quite simple, typical mycological methods or bacterial methods rather, of suspending plant material in a buffered suspension and then plating them out on particular media. And you can look at survival of these things quite easily at different levels within the crop. And you can find the presence of both Bacillus subtilis and amyloliquid quite easily. But does that tell you whether it's active or not? It doesn't. It just says it's there. It may not be actively growing. And indeed, with something like Bacillus, it may have just gone to its spore form and be really inactive. So it doesn't necessarily tell you whether it should be working or not. And then you've got the two fungal formulations there. So T34, I used to work on this many years ago, so I know it quite well. We've used this extensively in the past, and what we've actually seen with this one is that it does not survive in our particular substrate and with our particular crops. You apply it, you can find it in high numbers initially, and then it quickly dies out. Both that trichoderma and the glycladium there, you need them to be actively growing to actively, to again, act as competition, antagonism against other fungal pathogens, and for direct parasitism of pathogenic mycelium. So it's really, as Ant said, you really need to understand how these things work, and perhaps why they don't work in certain circumstances. I'm going to go on to a sort of specific application that we've been doing quite a bit of work on. So both the Serenade and Amylo-X have got approval for control of Botrytis on time, amongst many other fungal diseases. And we know they can work. They got their approval because they've been proved to work. But obviously our crop and our circumstances are different to a lot of that work. So there may be specific issues involved here. We all know what Botrytis looks like. There's a lovely picture there. But perhaps of interest in this case, and well, I'm going to come back to this, is if you look at that picture there, you'll see that Botrytis isn't on the sort of the canopy anywhere, it's on the stem, and if you look at the very early symptoms of the disease, you'll see that it starts right at the stem base, and that could well be important, which I'll go on to in a minute. We don't get much of this disease, it's very much winter specific. We just see the build up in numbers during the sort of shortest day period of the year. Doesn't really go above about sort of 8% of the crop at worst, and you can control most of it inevitably through the correct watering and environmental conditions. But we still do get a lower level. It's significant, and even very small amounts of disease right at the stem base. Again, if that product then gets sleeved, goes in a box through the distribution chain, then you'll get that disease developing through its sort of shelf life period. So it's really important to try and keep the numbers right down. So this is a bit of work we've done over the last 2 winters, looking at using either Amylo-X or the Serenade against Botrytis in time. And we've compared here the level of control we get with those 2 products versus a switch. So the fungicide switch, which was found is the most effective fungicide for this disease in this particular crop. So you can see the control level of disease where there's been no treatment. You're getting up to just over 7% there. Quite a quick sort of increase through the short day period, and then goes down again. And with switch, that's just a single application of switch. We played around a lot with the timing of when best to apply it, but just single application, and we're getting pretty good control. With the Amlo-X and the Serenade, we're looking at weekly applications. So sort of roughly speaking, a week before that switch, then at the same time as the switch, and then 2 subsequent applications. So that's an interesting point, which perhaps wasn't mentioned before, in that not only are some of these products more expensive, but you have to apply them more regularly as well. And that becomes a significant, not just in cost, but also in labour. So you can see both the Amlo-X and the Serenade really didn't give us any control at all. Serenade may be a hint of something earlier on, but really nothing, nothing to make you think, oh yes, let's use this product. That doesn't mean saying we're not going to carry on looking, and I think there are particular circumstances that mean that perhaps they don't work as well as we might expect them to. And like I say, we want these things to work, and we will persist in trying to get these things to work, and we're looking at getting an AHDB bit of work together with other pot herb growers to look at why they don't work for us. There may be specific reasons why these things don't work in herbs. Herbs are quite different, particularly in terms of like your volatile oil production. Is that volatile oil spectra interfering with some of the activity that we might otherwise expect? I don't know, this is just pure speculation, but this is really what we probably need to find out. So again, going back to these 2 sides of activity for the bacterial formulations, you've got these lipopeptides on the leaf surface. Are they there? Hopefully they are. Following on for the amber work, we've taken note of this application technology, and being very much aware of needing to get that right, particularly with biopesticides. But are those lipopeptides actually stable? Are the volatile oils of the herbs interfering with that stability, stopping them actually doing what they're supposed to do? That's quite possible. And also going back to the survival of the bacteria and the fungi, are they there? Well, you can find them quite easily, but are they active? And it's really, this is essential. They need to be actively growing to be able to outcompete, to be able to produce the sort of antagonistic substances they're able to produce, and to actually directly affect the fungal pathogens that might be there. And then, just as importantly, are we actually getting to the target? So going back to that picture of the Botrytis and the way I was talking about how it looks as though it's very much stem-based where that infection starts, and my own sort of way of thinking is that it could actually be some latent infection going on there, and there may even be some seed-borne disease there. So are you going to be able to achieve control in those circumstances where perhaps there's latent infection, particularly where there's seed-borne disease, that you're just simply not going to hit it with your biopesticide that are reliant on the sort of the surface infection by spores and mycelium. So it's really important to know not only just how these products work and whether they're working in your particular environment, but also how the disease is behaving, and therefore targeting your control of it correctly. So there's a lot of questions, I think, still to answer, certainly in our specific circumstances, and I would guess that everybody's a little bit different, and it's being able to think about how all these things work, how they all interact, as Ant was saying, and really applying them to your specific circumstances. So there's a long way to go yet. We want them to work. We're going to persist. We're going to try and get some work done to make them work. There are many other biopesticides we're also looking at, as I'm saying. There's things like growth stimulants. There are plant defense elicitators, all these other type of products, and we need to make them work. We need to understand how they work in our own circumstances. So that's just a very quick sort of overview of our experiences. We've not gone to work, but we're still trying. We need to ask a lot of questions to make it work. So that's me done. So if there are any questions, then I will attempt to answer them. My internet connection isn't the best, so sound is not brilliant. So hopefully I can hear anything that might be asked.
Thank you, Simon. Can you hear me okay?
I can hear you at the moment, yes.
Okay, good. If you just stop sharing your screen and then it's on the right hand side, there's a couple of questions. In this screen sharing. Don't worry.
You all right now?
So yeah, perfect. Thank you. Thank you very much for sharing. The work that you've done on your site. Clearly, you're very proactive. We've had a question in asking what your motivation is to do this, and obviously you touched on that. But I think that's obviously an element that needs to be considered when you're trying to get growers to work with these products and try and persevere and try different ways of using them and understand them better. Do you have any insight into what you think could be motivational, and what could stimulate other growers to sort of do the work that you're doing?
I think the immediate thing is that we are looking to use more and more things like your Bavaria bassiana products for the control of insect pests. So if you're also having to apply a fungicide of some sort, then that is probably going to interfere with your Bavaria. So it's essentially got an integrated approach that you need to be able to try and use something that's not going to interfere with other measures of control you've got going on. So that's kind of like the immediate thing. We want to be able to avoid those issues. And we are seeing more and more resistance. I mean, particularly with insecticides, but we do also see some resistance with some diseases now. So, longer-term vision, you think, well, let's start to make these things work, because longer term, we may not have various products, we may not have things that work. So we've got to make these things work now. There's still a lot to learn, so let's start learning now. So that's sort of my way.
Yeah. Have you looked into temperature when applying bacillus-based products? I understand that warmer temperatures can be key to success.
Yes, so again, we do have microclimate sensors within the crop. So we have a dashboard set up to show us if we got the right conditions for these things. So as far as the recommendations go, then yes, we are well within the range that these things should be working. We are, although I said that in the specific conditions I was talking about with Botrytis in winter, it's still a warm greenhouse. There's still plenty of temperature and the humidity there in order to make these things work properly. So yes.
Yeah. Another question for you. In your graph, there was already Botrytis present when you started your applications. What happens if you get them on as protectants before symptoms? However, they've said that they also agree that latent Botrytis is likely to be an issue.
Yeah, that's right. So, yeah. First of all, that data was only showing the interesting bit. There was a lot more before it, a lot after it. So we were starting well before starting to pick up that disease. So that's covered. And in a way, we started applying the biofungicide well before you start seeing the first signs of the disease. But as the questionnaire is pointing out, as I've said, that could be latent infection that you can't actually see at that point. And no matter what you do, unless it's something into the plant to stop that, then you're not going to achieve control. So that's something we need to understand better.
Yeah. Well, thank you very much. If there are a couple more questions, but I'll hold them until the end so that we can pass on to Dave for now. But thank you so much for your presentation, Simon. Dave is going to talk to us about practical ways you can manage the application of biofungicides to help try and improve integration into existing IPM programs and improve efficacy. So I will just pass over now. He's having some trouble with webcam, so unfortunately you won't be able to see his face.
That may be an advantage, actually. So can you see that okay?
Yeah, perfect. Thanks, Dave.
Brilliant. Right. So yeah, I'm going to take you through some of our work on primarily from the Amber project and talk about some of the kind of lessons that we've learned. So, oh, sorry, I should go back. So, so this is work done, as said, through Amber. So it's some of my work and work at Jill Prince at Warwick Crop Centre, but also work done by Claire Butler Ellis and colleagues at Silso on the spray application side, and Erica Wedgwood and colleagues at ADAS Boxworth. So just echoing some of the things that we've had before about motivations for use of biofungicides. So you're aware about the issues with resistance in plant pathogens. One thing you may not be aware of is a really serious issue that is coming through at the moment, and that's the increase in hospital-acquired Aspergillus and Candida infections. So these are infections in people which are resistant to antifungal medicines, primarily azoles and triazoles. And in fact, more people now die of fungal infections than they do from malaria or from breast cancer. And it's a very serious issue. And already, sorry, that noise is my automatic doorbell. I think someone's at the door. So already there are calls in the medical community to actually ban the use of fungicides, and certainly azole-based fungicides in horticulture, because this resistance is linked to use of fungicides by the horticultural industry. And it's a really serious issue. It's an issue that's not going to go away, and it's something we need to be aware of, because it's just going to increase the pressure on fungicide use, and somehow we need to actually turn that to our advantage and be able to leverage support from the government for using things like biofungicides as a public health good. So I think that's a route we could actually go down. So for the rest of the talk, in terms of talking about management of biofungicides, I'm going to emphasise a number of things. So one is the importance of understanding the mode of action of that active. I'm going to talk about two of the observations we've made on biofungicide use in amber, and then I'll talk about optimising spray application, and then finally some kind of work on integration. So the key point is that biofungicides can and do have multiple modes of action. So as Simon and Ant have been talking about a bit about this previously, you know, some produce antimicrobial compounds that kill plant pathogens, some compete with plant pathogens for space, some turn on plant defenses against disease, some are mycoparasites and they invade and kill plant pathogens, and some do all of those. So this cartoon here on the right is for trichoderma, applied to the root zone, and you can see it does multiple things. So it directly attacks pathogens, it colonizes the root zone itself, induces resistance, promotes root growth, and it primes defense responses in theory. Another key point, so I guess the point about that is that, and I'll come on to this in a minute, understanding that mode of action is important for being able to apply the biofungicide in the right way. The second thing, and the other speakers have touched on this, is that these are not like-for-like replacement for conventional synthetic chemistry, and as a result their intrinsic efficacy is lower, and as a consequence of that they work best with low to moderate disease levels. They're also, their activity is dose dependent, and so really to get the best out of them you need to maximize the amount of agent per unit area of plant, for example on the foliage. So this is just looking at the modes of actions of the different biofungicides, some of the different biofungicides that are available. So we have our preventatives for Botrytis, the bacillus products such as Serenade, and Simon said they work partly by the production of extracellular antifungal compounds, but also they can actively grow and form a biofilm on the plant surface to outcompete plant pathogens for establishment. Glircladium is a fast grower that also colonizes plant surfaces and can act as a hyperparasite. So these need to be applied before the disease is present so they can actually get that colonization in place. Others such as Amplomyces, which is in the product AQ10, that's an obligate parasite. And so there it's really an early curative, because it will, it has to have, it's a parasite powdery mildew, and it has to have powdery mildew there and in order for it to function at all. So timing is critical, and I'll come on to that in a second. And then finally, we have our root zone operatives. So again, anything applied to soil is really applied as a preventative, and trichoderma fits into that with its multiple modes of action. So these are some observations based from AMBER, and in AMBER what we've done is that we've worked with growers who have not used biopesticides before, and they've tried using, under our observation, biopesticides as part of their conventional crop protection programs, and then we've observed how they've got on with them. And from that we can start to learn lessons about why things may or not be working. So the first example is the use of AQ10, this parasite, on powdery mildew on cucumber, applied with a manually operated vertical boom. With that, we observed acceptable disease suppression on a Q variety with intermediate resistance, but no effective disease suppression on a susceptible variety, which kind of shows you the importance of the integrated nature of these products. We also picked up concerns about potential inhibition of the biofungicide by pesticide residues in the tank, and as a result of that we kind of recommend that people using biopesticides have separate tanks for conventional chemistry and for biopesticides. And then the other observation we made, which was common to all usage, was that the volume application was really high, and also we noticed uneven distribution on the crop. Second example, I know this is not a protected edible, but still we're flagging up. This is use of Gliocladium in pre-stop against Botrytis on cyclamen applied with a reaper gun. And this is an interesting example because in this case the pre-stop actually outperformed the fungicide standard, but in both cases control could be better. The volume was really high again, and I'll come on to this in the next few slides. This is 3,000 litres per hectare, which is really excessive. This is a data, so the plants with Botrytis in a standard fungicide treatment, 60% of plants actually had observable Botrytis, whereas with the biofungicide it was 28%. So this is an example of actually the biopesticide outperforming the standard chemistry. But as I said, you still want to go further and get better control with both of them. Okay, so Anne was saying, and Simon is saying, and I will now say, that the spray application is really a critical factor for all biopesticides. It's particularly because many of them have contact activity, and because so many of them are alive, they're not forgiving. If spray application is not on it, if it's poor, then they are likely to fail. And in general, we've observed that the water volumes being used at the moment are usually too high. And that means, firstly, that application is inefficient because it takes too long, and also that there is loss of product through runoff. In some cases, that the amount of runoff is really, really excessive. So to get best results, it's all about maximizing the amount of product per unit area of plant surface, which means using at the highest concentration you can within the label recs. And there's a common perception that actually high water volumes are best because you get better coverage and better penetration in the crop, but actually the truth is far more complex than that. This is data from Silso from their track spray experiments. This was done with basil, and they're using a dye as a kind of a proxy for pesticide or biopesticide active substance. And the graph shows on the bottom axis we have the volume, water volume, and on the vertical axis here, this is the total deposit on the crop in terms of microliters per gram. Sorry, my thing's going a bit funny. This blue line shows the total deposit, which you can see here. And it starts off that as you have increases in volume, you get more total deposit on the plant. which stands to reason because the more you apply, then the more that active will actually stick onto the plant. But after a while, it starts to plateau, and that's because you're getting runoff. So in essence, you might be applying more, but you're getting more runoff. So you're actually losing actives. So that there's no return by applying more volume. But look at this red line here. This is the normalized deposit. This is the proportion. So it's the amount of active substance per 100 litres of water. So as I said, it's the efficiency, if you like. It's the proportion that's sticking onto the plant. And now that shows the opposite trend. So there, the more water volume you have, the lower the proportion that actually adheres to the plant. And that's because you're diluting the product. And the most efficient zone is going to be kind of round about here, you know, about 500 litres upwards, where you're balancing out the effects of the blue line before you're getting to the plateau, and the effects of the red line where you're getting loss through runoff. Many of the recommendations for biopesticides are saying use at about 1,000–1,500 litres per hectare, which is far too high. So Silso have a series of recommendations based on their track spray experiments for horizontal application and also for taller crops. So this is from tracer dye studies on short crops applied with a horizontal boom. And in this case, the amount of active substance deposited on the plant is sensitive to the volume. So for a product applied at a constant dose, the maximum active substance is given by the lowest water volume, provided you don't exceed the max label concentration. And that's a really important point. For products applied at a constant concentration, the maximum volume recommended by Silso is about 1,000 litres per hectare. But for smaller plants, it's better to go down to 500 litres per hectare. But in most cases, you're actually using products applied at the constant dose model. For tall crops, pepper, tomato, where you're applying with a vertical boom, it's different. So there, the work we've done on tomato has shown that the amount of active substance deposited on the plant is actually insensitive to volume, which seems surprising, but it's actually due to the shielding effects of the leaves and the complex architecture of the foliage. So you don't get any gains by actually applying a higher volume. And in that situation, you're better off using a low volume if you're applying at a constant dose, because application is much more time efficient. So at a constant dose, go for the lowest volume you can. For product applied at a constant concentration, then the recs from Silso are that the maximum volume is about 1,000 litres per hectare. Silso have developed a really useful calculator, and it converts the volume needed for the vertical crop into the water volume required and applied to the floor area, because the label recommendations and regulations are based on a floor area basis, and that's available through our AMBER website, and it's something I recommend you to use. This is going back to this mycoparasite, the biofungicide AQ10, and again it echoes the point about understanding the mode of action. So this graph shows the number of experiment on tomato through AMBER, and it's a number of colonies on a vertical axis against time in the absence of the powdery mildew host. So the Amplomyces is applied to foliage, and then samples are taken every few days to actually work out how much living material is available. This goes to a point that Simon echoed about actually knowing whether stuff is actually alive. And you can see from the data that the Amplomyces only persists for 3 or 4 days on the foliage in the absence of the powdery mildew, because it needs the powdery mildew to feed, to survive, because it's a parasite of it. Why is this important? It's important because if you apply your AQ10 when there's no powdery mildew there, it will die. If you apply your AQ10 when there's too much powdery mildew there, then the biofungicide would not be able to control it because of its lower intrinsic efficacy. So therefore, timing is absolutely critical, which brings in the point about the importance for smart decision support system, which is Ant was echoing. And I really think these are going to be key in the future to helping maximise the benefit from biofungicides. Going to talk finally now about integration with conventional fungicides, because these biofungicides are not standalone products, and what we need to do is use them to reduce our use of conventional fungicides in their applications. There are knowledge gaps about compatibility of biofungicides with conventional fungicides, and that's something we need to fill. And as I said before, it's better to have separate tanks for conventional chemistry and for biopesticides to remove the concerns about inhibition of tank residues on biopesticides. There are reports in the scientific literature from researchers about co-application by tank mixing biofungicides and conventional fungicides. And these literal reports say that when you do that, you get superior control from actually using the fungicide on its own or the biofungicide on its own. And there are examples quoted for tank mixing trichoderma with conventional fungicides and for tank mixing the bacillus products with azoxystrobin. But what I'm not saying at all is going ahead and do this. I'm flagging it up to you because this is research from the literature, and I think what we need to do is actually check it out through doing our own experiments and actually find out whether it's applicable or not to our grower situations. And the take-home message from this slide is that we need to be using these biofungicides in a program to reduce the number of conventional fungicide applications in total, rather than looking at the biofungicide as a total replacement. This is data from Bayer from 10 years ago from the United States, in which they were trialling Serenade on table grapes for use against powdery mildew. Now, in this country, Serenade is used for control of Botrytis, but in North America, it also has recommendations for use on powdery mildew. So this is a graph showing leaf severity, so percentage leaf severity of disease against time, and the time is presented as number of days after application. This is the untreated control, and you can see how Botrytis disease develops over time, as you would expect. This is the progression of the disease using a standard calendar-based conventional fungicide application program, and the fungicide is giving you good levels of control. This is a program in which 2 of those standard chemical fungicide applications have been replaced by applications of Serenade, and you can actually see that you're getting effectively the same level of control by substituting to the fungicides with the biofungicide. And this is what happens when you do the same, but you don't have the Serenade in the gap, if you like. So it's showing you that the control is affected by the Serenade working in concert with the conventional fungicides, because if you remove the Serenade, you get an increase in disease progression seen here. And I think this is really important for going forward. So I'll wrap up now. I realise we're just slightly over time. So the conclusions are that we need to be using in the future biofungicides in an integrated program to actually reduce the total number of conventional fungicide applications. And what we want, and I think this is a message comes through from Simon very clearly, is we need measurable, incremental improvements in management practice rather than going for a giant leap, because we can't just go from using conventional fungicides to replacing them with biofungicides. It's not going to happen. It's going to be baby steps for all of us. So we need to combine not only biofungicides with conventional fungicides, and I know there are issues about those, but we need to view them also as part of a wider integrated disease management program, combined with cultural controls, environmental management, and smart decision support is going to be critical for that. Remember, these biofungicides work differently to conventionals. They're less forgiving, and as we've seen from the previous speakers, they require much more attention to detail. We need to take into account the modes of action. We need to consider compatibility, as Simon's been saying. And finally, good spray application is absolutely critical because the efficacy is dose dependent. We want to deliver the highest dose of product per unit area foliage for the things that are sprayed above ground. And in general, lower water volume is best because you get that higher concentration of product on the leaf surface. But we do need absolutely to have label reform and negotiate with CRD so that we can actually get on the label requirements the ability to use lower water volumes. So that is me. I'd finished. Just thank the people in that Amber team, and I'm quite happy to take any questions that you may have from me.
Thank you, Dave. If you could just stop sharing your screen, that would be great.
Yeah, I'll press in the magic button now. Right.
Lovely. I don't know whether Simon and Ant would like to. If you can't join us, unfortunately, via webcam, but if the others would like to, there are a couple more questions. We are a bit over time, but if you'd like to stick around for a couple more questions, you are welcome. So I've never heard of this, but have you tried to use EcoTac with biopesticides? Anyone heard of that?
No.
And anything from you?
Yeah, I have heard of it, but I can't remember exactly what it is. But we have not trialed it with our biopesticides. There may have been growers that have.
Okay, might have to come back to you on that one. So what effect of the efficacy of biopesticides should we anticipate depending on the water used for spraying, i.e. water quality, so pH? Aren't most growers now using mains water because of the fear of salmonella, etc.? Can treated water, even chlorinated mains water around 0.5 milligrams, have a negative impact on efficacy? Do you want to take Dave or Ant?
Well, both of us actually, but yes, it can. And water quality can affect efficacy. It affects mixing, for example. I mean, Ant is better qualified than me to talk about that, but it's something that really needs to be taken into account. And that underlines the need to actually, if you're adopting biofungicides into a program, to actually take these baby steps and start to trial it and to work with your agronomists and to be able to iron out these issues. Because what happens a lot is that people use a biopesticide and it doesn't work, and unfortunately we don't know the reasons for that, so we're not able to rectify it. That's one of the big mysteries with these products.
Yeah, I'll just pick up on what Dave says as well. In our technical notes on our products, so with the biopesticides that we bring through, we Chris in the office will put together the technical notes, which includes various protocols from the manufacturer that they've used in the trials, and that will have information on water quality, so what pH range it will work best at. And again, a lot of these questions that were covered by both the two other speakers, they're not ones at the moment that we have immediate comprehensive answers for. So in a lot of cases, it's going to be working alongside us. So if you're having issues with the products, get in touch and we'll be happy to work alongside. But it is important to access the literature that we have on the website, and if you're having trouble accessing that, then drop me or the technical team an email and we'll ping it across to you, because that'll have more details on the products in it.
Thank you. Are there investigations into toxins produced by EPF and their potential to be used to be the new chemicals of the future?
So I could,
yeah.
I could talk about, because I actually do research on it.
Yeah, go ahead.
So the entomopathogenic fungi, the EPF, produce a wide range of metabolites which have different impacts, and generally, yes, you are right, there are lots of opportunities to investigate natural enemies, if you like, for their natural products to be used as kind of rational pesticides, and I think it has a lot of potential because they should have higher efficacy and be more straightforward to use. But I would say that what we don't want to do is throw away the potential attractive properties of the biologically based control agents. So we would have to be concerned if we were using a metabolite about the evolution of resistance, because we could just be going down exactly the same road have been with chemical insecticides and stuck on the same treadmill, and we do not want to be doing that. You know, so I think let's take a measured approach to all of this. And sorry, I'll go off in a big rant as I always do, but we must not be looking for magic bullets. You know, we really must. We must. The future is integrated pest and disease management. And it's just having some vodka. But did you want to comment?
No, you've covered most of the points there. We've looked at it in the past around the metabolites produced from entomopathogenic fungus and the potential for plant protection products. There's obviously a couple of commercial issues in terms of the scalable production facilities to produce it. And then equally, like you say, the potential for resistance development is there. There's not many knocking around in the market that are looking to come to the UK as approved products, but it is definitely a technology that potentially could come further down the line.
Thank you very much. I think I'm just going to finish off with one more question. There are a couple more that are there, but we are over time, so we will get back to you one way or another. I'll share them with the speakers, and hopefully they'll provide insight on those. But just to finish off quite a big one, what would you think would be the most useful thing for us to understand to enable more effective use of biofungicides? There's no single one thing.
Well, I think I'll reiterate what Dave said in his previous answer, is that looking for a silver bullet or a nugget of wisdom, as it were, that's going to solve all the problems isn't necessarily a productive or possible thing to look at. I suppose openness and collaboration is the best way forward with this sort of thing. So obviously there's great growers like Simon and great researchers like Dave, bringing it all together, exchanging knowledge and information between groups so everyone's got the same level of understanding and can take that into their own environments to help develop the products, develop their efficacy in the field, that is going to be helpful across the board. So in terms of one nugget of wisdom that could solve everything, who knows, but definitely collaboration is key in these issues. Do
you have anything to add to that, Simon, as a grower?
Yeah.
Oh.
Yeah, absolutely. A key point really is having that knowledge available. So no disrespect to your ants, but these complex interactions and being able to understand how your disease and your biopesticide is potentially working. At the moment, it's difficult. Talk to somebody who has that knowledge for your specific condition. So really, it'd be great to see that being advanced in the future, having that sort of real expertise to work with, because at the moment it's not quite there. So that's how I'd like to see it go. Yeah.
Dave, last point.
Yeah, I would something that's left field, but it picks up on the point that Ant was making, is that growers need support because you are being asked by the government to radically change how you grow and to do things which are a public good, because reducing reliance on conventional chemical pesticides is a public good, and therefore you have a right to be supported in that. And and I would really like to see the grower community lobby CRD and the government for support through the National Action Plan. And this thing about the fungicide resistant opportunistic infections in people, you have to say, look, if you're going to ask us to reduce fungicide applications in order to protect lives of people in hospital, then you've got to pay us to do it. And that they do that in the United States for the IR4 program. So if we're going to transition to this new way of growing and protecting our crops, you can't just be expected to get on with it. You know, you need support from central government. So I think really got to lobby hard for that. It's absolutely critically important.
Thank you very much. So on that note, I will wrap up. I hope you can see my screen there. So yeah, thank you very much to Ant and Simon and Dave for their really useful presentations. If there are further questions, please do send them to me via email, and I will get them to the presenters and we'll get them answered for you. To remind you, the webinar has been recorded and should be made available shortly on the events archive web pages. And also I recommend that you make the most of other upcoming horticulture webinars, which you can register for via the events page. So with that, thank you very much for listening. I hope you have a good day, and goodbye.